This video looks at the Advanced Color Distance Analysis plugin. To start with, let's look at an image that is set up for use with this plugin. For this, we're only going to be examining one single image and not multiple images that will follow in a subsequent video. What we have here is an image that has been what I would call softly masked, in that while it looks like it's thresholded, it's actually still got all its color information. What we've done is use the brightness and contrast tool to set a line. So you can see with the cyan channel, it's still actually all there. And all we're doing is we're setting that threshold there, and then we're not clicking apply to make that actually binary. It's because we're actually going to use all that information. So you do this for every channel. You have a final channel, which is a mask. For this, the order we have is that the mandible is region 1, molar is region 2, the incisor alveolar 3, molar alveolar 4, nothing in 5, and then incisor 6. Once you have an image set up like this, you can start the plugin. Now, it's set up for an experiment that I've done. It should save your preferences, though your mileage may vary. We have one section and one sample. That's because it allows you to have not only multiple sections from the same series, but multiple series from different animals. We have 4 colors. I'm going to set the color sampling range to 5. We're going to do 10,000 iterations. We're going to use clustering method average, matrix composition method average. If you're doing only one side of an animal or a sagittal section, you want to uncheck this box. You can leave everything else on. We need to change this to manual. We're going to manually set this image. We haven't saved it, and then directory means it will just load it from a folder, but manual, we can just pick the image we want. Then we need to put in some names for the regions. So we had mandible, molar, incisor alveolar, molar alveolar, nothing, and incisor. Note that I've done them each on one line. Once I've done that, I can click OK. If you're on manual mode, this is what you'll see. Now, because we're only doing one section, it just says sample A, section A. We can ignore that, and we just take the image that you want to use, click measure current image, and finish. Then you wait. What you'll get out is all the trees that you've made with the scoring annotation and without. You'll notice that they're different, but some of them are very similar. And you'll also get out 4 consensus trees. You want to save both of these things because sometimes it is useful to come back to these and look at some trees, some individual trees that had a high score, or so that you can better understand what's going on in your consensus trees. So what you can do is then copy this and take it, copy a line out of this and take it into your tree drawing software of choice. I recommend ITOL, Interactive Tree of Life Online. And just to remind you on what these different terminologies mean, concordancy is similar to a bootstrap. It's not quite a bootstrap. What that means is it's constructed out of the clusters, the groups of leaves that were present in the most commonly found in every tree. And similar is a similar idea, except instead of looking for whether a cluster is exactly there, clusters get a score based on their the relative similarity of the cluster present to the one that you're looking for. So if there's like a 75% overlap, it will get a 0.75 score. And consensus, the straight consensus is the concordant consensus and is the best one to use, but the others can provide some insight or something to discuss.